Hello, and welcome back to Introduction to Genetics and Evolution. In the previous video, we talked about looking for evidence for natural selection at individual genes and trying to focus on differences, say, between humans and chimps and infer whether these differences we see may have been the result in part from the action of natural selection versus genetic drift. DNDS test is useful in this regard. This is looking at the number of non-synonymous changes per non-synonymous site divided by synonymous changes per synonymous site. The advantage of scaling with the synonymous changes is these are expected to be neutral, whereas the non-synonymous changes may be advantageous, may be neutral, or may be detrimental. The problem, though, is DNDS can sometimes mix up constraint with adaptive evolution and just call it neutral. So this is a little bit of a challenge, and DNDS in general is thought to be fairly conservative when looking for adaptive amino acids. When I say conservative, I mean that there's too many false negatives. There's too many times when you fail to see positive selection that actually has potentially operated in the evolutionary history of a particular gene. Now, we can leverage some aspect of neutral theory that we haven't talked about yet, and that is that we predict the ratio of non-synonymous to synonymous changes should be roughly constant through time, right? That the ratio that you see among individuals within species should be roughly similar to the ratio you see between species. You may be wondering, why should this be constant through time? Well, there's an assumption about the neutral theory that most non-deleterious, non-synonymous mutations are neutral. Basically, those mutations that you see within a species that are just segregating around, that they are at some frequency, those tend to be neutral because the bad ones are eliminated, the advantageous ones spread. So you tend to see a lot of these neutral ones. So these non-deleterious, non-synonymous mutations will behave like synonymous mutations, and they'll rise at some sort of constant rate, and they'll fix with about the same probability as a synonymous change. That, this idea of the allele frequency should predict 1 over 2n. Now, two fellows, John McDonald and Marty Kreitman, suggested this test, which has subsequently been named after them. They didn't call it the McDonald-Kreitman test, they just named it a test. Now, they proposed that you can test for selection by seeing if this ratio is constant within species and between species. The importance here is this within species is looking at the present day. When we're taking a snapshot of a population right now, what do we see in terms of non-synonymous variation, synonymous variation? The between species indicates historical changes, what's happened over the evolutionary history. So what they do is they align sets of sequences and identify the variable nucleotide sites. And they basically just have this 4-cell test where they identify each difference as a non-synonymous difference within species, a synonymous difference within species, a non-synonymous difference between species, or a synonymous difference. So let's try this. That was a lot of words. Let's try it in practice. So here we have an example. Let's say we have 4 individuals of species 1 and 1 individual of species 2. For the sake of argument, let's say species 1 are humans and species 2 are chimps. Now here's our codon table again. And what we're looking for are these 2 parallel categories. First, what is fixed between. So these are differences between species 1 and species 2. We have polymorphism within, so basically what is variable within, in this case, species 1. And then we can also categorize any of these differences as being non-synonymous, or amino acid changing, or synonymous, where they don't change them. So what we do is we just walk through. In this case, we're not looking per site, we're actually just looking overall. So where are the differences between species here? Well, here is a difference, or within or between species. So there's a difference here between species. This is within species. This is between species. This is within species. This is within species. So we have an amalgam here. We have some within species differences, some between species differences. Okay, now let's walk through them. So this first site here, this is a non-synonymous change. As you can see here, a CT anything to ATG, which is methionine. That is a non-synonymous difference, and this is a non-synonymous difference between species. So that goes in this cell. We skip the next one because we're not going per site. We're just going to skip the next one and go to the 3rd nucleotide site. In this case, we have a difference within species, but they all are making leucine. So this would be polymorphic within, right, and synonymous. So let's jump now over a little bit. This one right here is a difference between species, but again, AC anything is threonine, so this is a synonymous but between species difference. Jump over a little bit. This is a within species difference, but second position. Second position is always non-synonymous, so this would be non-synonymous within. And the last one is over here, ACC or ACA, they're both threonine, so this would be another polymorphic within species, because the variation is just in species 1. Species 2 has one of the ones from species 1, and it is synonymous. So we basically tally these things up, And we have these four numbers that come out. We'll just call them A, B, C, and D for the sake of argument. If all non-synonymous differences are neutral, okay? If all non-synonymous differences are neutral, then we expect A divided by C, this ratio that you see between species, this is essentially reflecting the historical, should be equal to the ratio of B over D, what you see within species, or the present day. In contrast, if some, or especially if there's a lot, of non-synonymous differences between species that were advantageous and selected. Let's say something helped us humans be able to digest things that are unique to our diet better. Then this ratio should be greater than this ratio. That the A over C should be greater than B over D. Okay? That what's happened then is these differences will have spread within humans. They are changing the amino acid variation. Since they spread within humans, they are now a between species difference. They're not something that's neutral, that's just kind of hanging out in the population. Okay, so let's look at a couple of examples. Here's some actual data from a few genes. Here's SEMP1, that enables cellular survival under low oxygen. This is a case where we see a large difference in terms of non-synonymous over synonymous between species relative to within species. This would be a good candidate for positive selection. There's been some sort of adaptive difference between humans and chimps. In contrast, this one, CIAS1, which affects autoinflammatory response, that one is pretty similar between the two. So this is a case where we can't reject neutrality. The ratio here of 5/10 is equal to 1/2. Now what about the opposite? This is one we haven't talked about. What happens when, instead of being greater between species or equal, what happens if it's the other way? So here's the gene AGT. The mutations in AGT affect hypertension. They're associated with coronary artery disease. What happens if you have a high number of non-synonymous differences among humans, a higher ratio of non-synonymous differences among humans than between humans and chimps? What do you think that might mean? So now we have to think about what kind of variation would do this. So what kind of variation persists for a long time but doesn't fix? Well, the answer in this case is unfit recessive alleles. So let me show you a picture. And this is a picture you saw before. Let's say over here on the x-axis is time in generations, and on the y-axis is allele frequency. Now let's imagine hypothetically that you had an allele that was actually bad, but it was recessive. What you might see something that looks like this. We saw this actually in previous videos when we talked about the action of natural selection, that it's actually kind of hard to completely get rid of these recessive bad alleles. These bad alleles can stick around for a long time because what's happening here, let's say for example the bad one is little a, but then the good ones are big A, and big A, little a, right? What happens as it gets very rare, almost all the little a's are present in the heterozygous form. So they're basically masked, and selection isn't getting rid of them very well. Only the very small number of little a's are actually being eliminated by natural selection. So the better dominant allele never quite gets to fixation. Now, a lot of diseases, as I mentioned way back when, tend to be recessive. And as a result, these mutations causing disease will stick around within populations, but never fix, and they eventually do get lost, but it takes a very long time for them to get lost. So this is variation that you'll see that will stick around within species, and it affects this within species numbers, but does not affect the between species numbers. And these diseases will tend to be associated with non-synonymous bad variation. So this is what causes that essentially, there's the ratios again we talked about, between species non-synonymous over synonymous versus within species non-synonymous over synonymous. That if all non-synonymous differences were neutral, we expect these ratios to be similar. If some of these non-synonymous differences were advantageous and selected, then we expect this ratio, A over C, to be greater than this ratio, B over D, right? Because the advantageous variants would spread. They would become between species non-synonymous variation. In contrast, if you have maladaptive non-synonymous differences, they will persist within species and never be converted to between species differences. In that case, A over C will be less than B over D. That's what will happen if you have diseases lingering around. So, looking at the McDonald-Kreitman test, we have this A over C equals B over D. That's a case where you can't reject neutrality. Again, it doesn't mean it's neutral. It can be a mix, but you can't reject it. If A over C is greater than B over D, then we have increased non-synonymous changes between species. We have more non-synonymous changes than we expected between species relative to within. And that is indicative of positive selection or advantageous variation. So you're running. In contrast, if A over C is less than B over D, then you have decreased non-synonymous changes between species. What that means is you have some variation that's lingering within species and doesn't quite get fixed. That is often the result of negative selection. There are other possibilities. Things like overdominance that we talked about earlier will also do this same sort of pattern, but it's thought that overdominance is not especially common, so a lot of this is going to be in this boat of negative selection or bad variation sticking around. So what happens when we look at the genome? I showed you a couple of examples. This is looking at a lot of genes studied across, or differences between humans and chimps. The blue marked genes are ones that have McDonald-Kreitman tests results indicative of positive selection. The red ones are indicative of negative selection. You notice there's a lot more red out there than blue. But again, we know that most mutations are bad. We know that most bad mutations are recessive. Therefore, it's going to make it so they're just kind of lingering around. So instead of just looking at this figure, let me give you some of the numbers. The positively selected genes from that particular study I was just showing you, there are about 304. Again, what are they associated with? Same sorts of things as the high DNDS values. Immunity protein genes, gamete formation ones, also some for sensory perception. In terms of negatively selected ones, many of these are involved in cytoskeletal formation, and they're often associated with diseases like muscular dystrophy, cardiovascular disease, etc. So let me give you one to try here, so you can have a little bit of practice within the video before you even try any of the practice problems. So this is a hypothetical sequence for ARSD. This is something that controls arsenic resistance. Okay? So I want you to go through here and calculate and interpret the McDonald-Kreitman test. So you may want to pause the video before you actually get to the in-video quiz. Okay, well, I hope that wasn't too difficult for you. Let's go ahead and take a look. So what I'm going to do is I'm going to highlight things in blue and red first. So the blue ones here are indicating differences between species. So this is comparing humans to chimp. The blue ones are indicating differences between species. The red are indicating sites that are variable within species, just to make it a little bit easier to see. So let's look at those. When we're looking between species, we have one non-synonymous difference. That's this one right here. This is non-synonymous. And we have two synonymous differences between species. Right? So this is a synonymous difference here. This is a synonymous difference here. When we're looking within species, what do we see? We have two non-synonymous. This one is non-synonymous. Remember, always the second site is non-synonymous. This one's also non-synonymous. And this one right here is synonymous. So one synonymous within species. So our ratio is 1 to 2 versus 2 to 1. So again, which one is bigger? In this case, we see it's bigger in the within species category. That this is the result in the direction of negative selection, that there is potentially bad variation sticking around within species not being eliminated. So let's recap real quickly. Here's the McDonald-Kreitman test. Again, if A over C, A being the between species non-synonymous divided by the between species synonymous for C, is equal to B over D, the same for within species, you can't reject neutrality. Does not mean that it is neutral. If it's greater between species than within species, then you have increased non-synonymous changes, that interesting positive selection, right? In contrast, if you have A over C less than B over D, you have decreased. You've been holding back the non-synonymous changes. They're staying within species and not converting to differences between. So that is negative selection. Now, I've shown you the DNDS test and I've shown you the McDonald-Kreitman test. There are other metrics out there. There are many that also exist that we won't use in class. The one popular one is referred to as Tajima's D. That uses the frequencies of alleles to test for recent selection or recent changes in population size, as an example. But importantly, these are implemented to understand what's going on. There's two general approaches for studying. One is to scan the whole genome using these metrics and look for possible selection and then try to interpret it. Right? So that's like the picture I showed you there showing all the red and blue labeled genes. That's when we're just looking at everything and trying to interpret how much positive selection versus negative selection is there in the genome. The other is to look at candidate genes, to pick out the subset of genes that you know affect brain size or affect speech, and look for the signatures of selection at those, or maybe even individual genes. Now, both of these are active areas of research, and both of them have given us some clues as to what's been important in our divergence or the divergence of any other species that have been studied. So closing out, what makes humans special? Well, these DNA sequence-based tests have shown that, again, hundreds of genes have undergone selection to change, and a lot of that seems to be, when I say selection to change, I'm referring to this positive selection, these adaptive variants. More genes are under some selection to stay the same, that basically non-synonymous changes are bad and therefore they're held back. They don't ever become differences between species but may linger as differences within species. and therefore some of these bad alleles are sticking around. And this also makes it so a lot of our sequence is more similar to chimpanzees than if we were evolving purely neutrally. But importantly, let me just close with this, there's no single gene change that made us what we are today. Yet, we're not as different from chimpanzees genetically as we might like to think. Well, I hope you found this interesting. Thank you for joining us.